Greetings and welcome to the next webinar on riparian ecology and management. Today we have Dr. Cummins. He's a stream and watershed ecologist specializing in aquatic ecosystem processes with emphasis on freshwater invertebrates. He has authored over 100 peer-reviewed research papers and is an editor and contributor to the book Aquatic Insects of North America, now in its 4th edition. With his colleagues at Humboldt State University and the Forest Service Pacific Southwest Station, he continues to conduct research on the role of aquatic insects in endogenous salmonid productivity. So with that, I'll turn it over to Ken.
Thanks very much, Richard. As you can see from the title, the theme of my presentation is that the potential for management options in the riparian corridor along forested coastal streams in Northern California involves management to improve salmonid production. That seems to be the major goal, and that's to accomplish through maximizing the availability of key macroinvertebrate prey organisms for the salmonid populations. That's the major thrust of my presentation. The riparian, as well known, the riparian zone along streams in forested watersheds has a lot of functions related to how the stream ecosystem functions. You can see they're both physical and biological interactions. The ones I'm focusing on in this talk are the shading by riparian canopy cover and the litter inputs from the riparian plant species. So that the management assume it will be a combination of managing for changing the relationship of the shade and the species composition of the trees which influence the litter inputs. The assumptions that are involved that I'm using these following assumptions. It's probably good to state these out at the beginning. First of all, as I just mentioned, I believe that the management of the riparian zone will involve a combination of thinning the canopy to increase light and the manipulation of the species of the trees in the riparian by selective harvest. And secondly, the most informative measure, I think by consensus among stream ecologists, the most informative measure of ecosystem function is PR, and a notation PR, and that's the ratio of autotrophic production, that is plants in the stream, and community respiration, that's the decomposition of all the organic matter in the stream. We can simplify this for this talk. The P then represents basically algal production, or algae in the stream, and the R represents the utilization and metabolism of litter from the riparian zone. So it's algae, P, and litter, R. Those are the two things we're going to manipulate, presumably, in management. And this notation I'll just use as throughout the talk. As I said, knowing this about a stream ecosystem seasonally or annually, most stream ecologists, I think, would agree this is probably the most fundamental measure you could make. It's difficult to do, and so it hasn't been done very often, or when it's done, it's in very short time periods. So there's a lot of calculations and modeling that have been done to try to work this out, but it's a fairly simple concept, but difficult to measure. The third one is that the macroinvertebrate food is as important as habitat in supporting stream ecosystems function, and the ones that we're considering here are the invertebrates that serve as food supply for the salmonids. I think the general feeling among fisheries people, or at least their stated feeling, is that if you get the habitat right, the fish will be fine. My caveat for that is that, you can make the habitat look good or work well, but what are they going to eat? You have to remember that the best salmonid production ever measured is in places where there is no habitat diversity whatsoever, and we call these hatcheries. So in a hatchery, it's all about food. And I think in streams, it's at least 50% all about food. And the fourth one is that I'm assuming that the measure of the success of any management plan involving the riparian zone, in which salmonid populations are the ultimate target, will be specific growth rates in the population, as the specific growth rate per individual, and then averaged over the population as a measure of how well a salmonid population is doing. Okay. Now, just to quickly on the PR part, The P is the in-stream plant growth, and we're using primarily algae in forested streams, and the total community respiration. Really, the measure here is the rate at which the litter that comes from a riparian zone is processed or turned over. In other words, that organic matter incorporated into stream processes. If the PR is greater than 1, then basically the algae are winning. If the PR is less than 1, basically the litter is winning in terms of the driving the energy transfers in the stream ecosystem. So if it's equal, they're balanced. Greater than 1, the algae are dominant. Less than 1, the litter is dominant. And of course, this will have a significant seasonal component to it, particularly if we're watching deciduous riparian plant trees. I might just point out that PR less than 1 is certainly the dominant condition in forested streams in all of North America, probably worldwide as far as that goes, but certainly in Northern California. Okay, so here's a slide that's not very easy to read, but I think the concept is simple. This is data from a watershed in Michigan, and I'm using this because we don't have similar data from streams in Northern California, probably on the Northwest in general. On the y-axis, you go from low to fast, slow to fast, low to high processing of the litter once it's in the stream. And on the x-axis, we have the PR, where PR of 1 right here is the balance between, this is PR greater than 1, the algae are winning, PR less than 1, the litter's winning, sort of. And these are different size streams along the watershed, from a first-order stream all the way down to a fifth-order stream down here. And you can see when litter processing is high, PR is less than 1. So the system is dominated by litter inputs. When PR is greater than 1, litter processing is slow. And you get a very nice relationship in this watershed with lots and lots of data being collected to demonstrate this fact. So there is a difference between whether the algae are driving the system or the litter inputs are driving the system, and that's certainly going to be important, I believe, in deciding exactly how to manage the plant system in the riparian. Just as some information, background information as to how we have come to this point of believing that light and litter are important. Stan Gregory at Oregon State did some really neat experiments early on in the 70s, late 70s, and he took paired streams and old growth streams in the Cascades, Douglas fir primarily, and he selected some streams to add significant amounts of nitrogen and phosphorus. These streams, like the streams in Northern California, are extremely low in nitrogen and phosphorus, microgram levels. So there's very little nutrient in these streams, and so he gave large doses of nitrogen and phosphorus to the old growth streams, in some of the old growth streams, and there was absolutely no algal response. But with no nitrogen and phosphorus additions, he brought a generator in and turned the lights on, and within several days there were big algal blooms in the stream. So it's a clear indication that light and not nutrients are limiting in these low nutrient streams. And that includes, it hasn't been tested here as he did, but I believe that that's certainly the case in Northern California. So the other thing is that when Stan added nutrients plus lights, then he got an increased algal response, but it was filamentous blue-green algae primarily. And I just point out that from an invertebrate point of view, the filamentous algae is a really poor substrate for invertebrates. Very, very few invertebrates feed directly on filamentous algae. It may offer some substrate for habitat, but it's certainly not very conducive to maintaining high diversity invertebrate populations in streams. So it's sort of be careful what you wish for. You get a lot of algae, but you don't want to also involve any sort of nutrient enrichment because it'll drive the system to filamentous forms. Now, in the early part of the 2000s, Mundy and his workers in Europe enriched substrates in streams with wheat grains, and showed that he could increase the density of the invertebrate populations 1.5 to 3 times over the controls. And their general conclusion was that this non-algal organic food could be added to streams, increasing salmonid food and presumably increasing salmonid production. So you can manipulate the algae with light, but you can also manipulate the organic matter in the stream by just adding it directly. As far as the importance of food, Wilsbach, in some field and laboratory studies, some really nice studies, I believe, used a food delivery tube system where he could deliver food to a location in the stream remotely so the fish couldn't see you. And by changing the location of the food delivery, you could easily change the location of the fish relative to their habitat. And she repeated the same kind of experiments in lab channels where she provided habitat and food delivery by the tube system and was able to move and maintain locations of fish relative to the food availability. She simulated predation with a battery-driven small scuba diver that swam around and scared the fish, but that didn't seem to have a lot of effect. They still tracked the food. So just some evidence from the field and the lab that food is probably pretty key. In a real large experiment done by Wilsbach and Harvey at Humboldt State, Wilsbach with the California Cooperative Fisheries Unit, and Harvey with the Forest Service, on green diamond land. Green diamond has been a big supporter of this kind of research from the beginning. They removed 100-meter sections of canopy, mostly red alder, and what they observed was an increase in salmonid production growth rate, and assumed it was related to an increase in macroinvertebrate production that resulted from greater algal densities resulting from more light. That's an assumption. That part wasn't tested, but they certainly had strong correlation between more light and better salmonid growth. Just a diagram to simplify the general relationship. I just wanted to put this up. Here's the light, which is driving this part of the system. The major invertebrates that are responding to that are called scrapers because they feed primarily on the algae that develops on the surface of rocks, for example, in the flowing water. So the light, get the arrow here. The light is the energy driver causing algae to grow and then fed upon by invertebrates who are adapted to remove that rather tightly attached algae from rock and cobble and gravel surfaces in flowing water. Here's just not a terribly great picture, but this is a really common mayfly family of scrapers that occur in certainly all streams in non-polluted streams in Northern California. It'd be very easy to find this particular group of mayflies that make their living by removing the algae. You can see here's an experiment that was done. The white tiles were colonized by algae, tightly attached algae, and then a caddisfly. This is the caddisfly here in the stone case, and where they've grazed, you see they've removed the algae, and you can see the tile. You can see how efficiently these animals can actually graze. Notice that the grazing paths are circular. This makes some evolutionary sense because for these animals, the world is in fact flat, and you can fall off the edge, and it's probably better to maybe do a little overgrazing but stay on the rock. The next two slides actually show some examples for that particular stone case caddisfly that's shown in the slide before. This is just gut contents from the 4 larval stages of that particular animal, first, second, third, fourth, and fifth instars, and then averaged over the life cycle. The white is algae, the black is detritus or dead organic matter. You can see that in this particular population, the feeding was dominated by feeding on algae for sure. Here's the same species in a different stream. This stream, by the way, is a PR greater than 1 stream. This stream is a PR less than 1 stream. And you can see that, again, over the 5 larval stages and averaged over the life cycle, the dominant food that the animals were scraping up was detritus and not algae. This would be organic matter that was settling on the surface of the rocks. And I should point out that the final stage of the larvae in this particular population was the weight was twice the weight of the final stage of the larvae in this population. So getting algae for this animal has major repercussions probably for adaptation and survival of larger females being produced, for probably producing larger animals in the next population. So that's sort of the algae side of the picture. We know a lot that light is what's limiting primarily, and that if you increase the algae, it goes primarily into the group of invertebrates we call scrapers. Many different taxa involved in that group, but they all do the same thing, namely they're adapted to scrape algae from surfaces of rocks in the stream. What do we actually know about the other side of the PR, the R part, the litter part? A number of things. There have been studies going on this part of the story probably for 40 or 50 years now. You measure the percent riparian canopy cover by different tree species in the riparian. Just get the percent cover in the riparian zone. That predicts very accurately the percent composition of the litter that you could find retained in the stream. That's not surprising, but it's nice to have empirical validation of this, because if you have measure this cover, which you can do remotely with GIS, then you have a good predictor of what's going to be the composition of litter in the stream. Once the litter is retained, it loses a lot of its weight, 30 to 40% of its dry weight in 24 to 48 hours. The litter becomes a good carbon source, but a poor nitrogen and phosphorus source, because most of those soluble compounds are rich in nitrogen and phosphorus. The species of riparian tree or shrub that's in the riparian is a very accurate predictor of how fast, based on temperature, the litter will be turned over. There's fast litter and slow litter depending on the species of riparian tree. So if you want carbon to be cycled quickly, you would want to maintain the fast processing litter stream species like red alder. If you want the litter to be processed more slowly, then you would want to maintain the slow processing litter species like redwood, tan oak, and some of the others. So we know how fast these things are processed, and the best way to describe this is on a temperature-dependent basis. That is, if you measure the median temperature each day during the time the processing is going on and sum that over the period, those are degree days. So, for example, 10-degree temperature in a stream for 10 days gives you 100-degree days. This allows you to compare cool and warm water periods, because you want to know the amount of processing per degree day. Of course, it'll take longer at low temperatures, and it'll go faster at high temperatures. So just the degree day procedure is a useful way to keep track of temperature in terms of how fast this litter is going to be processed and converted in the stream ecosystem. So the processing involves, first of all, the colonization of the litter by microbes. They enter the litter and begin to metabolize it. They actually turn this very rich carbon source and poor nitrogen phosphorus source into a good nitrogen phosphorus source by virtue of their own biomass accumulation. Then selective feeding by shredders occurs on the most conditioned litter, that is the litter with the most microbes. And that all these processes together with fragmentation have the overall effect of converting leaf litter or plant litter from the riparian zone, or coarse particulate, or get the arrow again, coarse particulate organic matter, like the litter, to fine particulate organic matter, or small particles. And those small particles, which are generated by this process, are further utilized by microbes. I mean, the FPM is further used by microbes and shredders and collectors. This, by the way, this should be collectors. We'll get back to that in a second. Okay, so here's the diagram that summarizes that. The leaf from the riparian enters the stream. It loses a lot of dissolved organic, 30 to 40% in 48 hours, pretty much independent of temperature. So that's the tea bag effect. You put a tea leaf in a cup of cold water, and of course it leaches. The same thing happens to the leaves. Here's a leaf going in. Once the leaching is completed, or pretty well completed, the microorganisms invade the leaf and begin to metabolize the leaf. And then once the microbes are in the matrix of the leaf, the shredders begin to feed on the leaf and grow and produce more particles, especially their feces. The analogy that's been used is the leaf is like a cracker. The microbes are like peanut butter. The shredders require and use peanut butter, but the only place peanut butter happens is on crackers. And the major biomass of the microbes is in the matrix of the leaf. So for the shredders to get that, they have to eat through the tissues. And the output of this, the major output of this is, so leaf leaching, microbes in, shredders feeding. The major output is fine particulate organic matter, which feeds, the major group that it feeds are the collector invertebrates. And it's among the collector invertebrates that are the overwhelming majority of those invertebrates which are consumed by juvenile anadromous salmonids and probably resident salmonids. Okay. Okay, here's a picture of the peanut butter, scanning electron microscope shot and a light microscope shot across the surface of a leaf here. And these are all hyphomycete spores. The hyphomycete fungi are really the critical component of the peanut butter. And that's been demonstrated. For example, if you extract the lipids from these hyphomycete fungi and spread it on filter paper, the shredders will eat the filter paper, which of course they do not eat if it hasn't got the lipids from this particular group of organisms. So that's what the shredders are searching for and feeding on in the litter. Here's a picture of one of the stonefly shredders. It's quite large. You can see from the scale that these stoneflies, when they're fully grown nymphs, reach almost 2 inches. So they're very large. There's an eastern species, which you're looking at here. There's also a western one that we have that's quite abundant. And you can see this is a handful of leaves fed on by these 5 stoneflies for about 48 hours in culture. And you can see they've reduced the full leaves to major veins and midribs. And those parts of the leaf, of course, have more lignin and less microbe colonization at the point. So you can see that they are selectively feeding on the portions of the leaves that have the most fungal biomass. Okay, here's 3 different leaves that were left in the stream for the same length of time. Here's a red alder up here, red alder here. Here's a maple here. This happens to be a vine maple, but a big leaf maple is probably more common in Northern California streams. And here's rhododendron. These are all in the stream at the same time. Here's a caddisfly shredder, which is feeding on these leaves. And you can see that the red alder was pretty skeletonized. The very, very fast, probably 2-week time here, I think about 110 degree days. And here's the maple, which was fed on slightly slower, but still pretty fast. And here's rhododendron that looks like a piece of shoe leather, and it has certainly not even been colonized effectively by the microbes yet. So the rates of turnover are drastically different between the fast ones and the slow ones. And so that has to be taken into account into how fast you want the carbon to cycle in a stream by managing different plant species in the riparian zone. Well, the shredders are real shredding mills, and they take leaves and turn them into feces, basically. This is a handful of leaves, alder, I believe, fed on by one of the shredders, a crane fly shredder in this case, and these are all fecal pellets. And it's fairly significant. These animals feed at about 150% of their body weight every day. That's pretty significant considering that we're somewhere less than 2%. So that's a major turnaround from coarse particulate leaf to fine particulate feces. And you can see again that they've left behind the major veins and midribs, which are are fairly rich in lignin, which is quite resistant to microbial decomposition initially. Okay, the rates at which the litter is processed has been studied probably for almost 45 years now in the field using a very simple bioassay. As you make your own leaf packs, leaf accumulations, weigh them and put them on an obstruction here, a brick, and set them in the current in the stream so that the current is actually going this way and striking against the leaves as it would against a rock or perhaps a piece of wood in the stream. You can do this with either broadleaves, or you can do this with needles. Here some are strung on a monofilament. And so that gives you a measure in the field, a simple bioassay measure in the field of the rate at which the litter is disappearing. And also, you can, of course, collect the shredders that are accumulating on there and actually feeding on them. We've used this simple bioassay in stream ecology classes, at least I have, and many of my colleagues have, for over 40 years now. It's very simple. We've done it with grade schools, graduate students, research projects. Even university administrators seem to be able to manage this particular experiment. Okay, so here's some actual data from this kind of experiment. On the y-axis is the percent of the leaf that you put in the stream that remains, and this is degree days down here. Remember that for example, 200 degree days would be 20 degrees at for 10 days, or 10 degrees for 20 days. And you can see that if you look at when about half of the leaf litter that went in, that is after leaching, has been converted to small particles, it's about 250 degree days for a track of fast litter, and it's about 600 degree days for a track of slow litter. In this particular case, I think this is alder, and I believe this is probably tan oak. So, and the R-squares, which are off the graph, are all reasonably good. So those are fairly good fits, and from large data sets. So if you know the temperature, median temperature for the day, can add up to degree days, and you know the leaf, and you know along here, and you know the leaf species, then you can be a very good predictor, very accurate predictor of how fast that material is being utilized in the stream ecosystem. Okay, so a big set of studies done by Wilsbach and Harvey, again sponsored by Green Diamond. In this experiment, they increased the light by removing red alder canopy along 100-meter reaches, and that correlated, as I said before, well with increased salmonid-specific growth. In addition, another part of this experiment was that they added high densities of Chinook salmon carcasses. The conventional wisdom in this area is that one of the problems with the loss of salmon migration that was the mechanism by which marine nutrients were returned to the headwaters by the salmon coming back from growing up in the ocean and dying in the streams. And so the assumption now is that those salmon carcass nutrients are missing. There's certainly stable isotope data showing that, qualitatively at least, that those marine isotopes show up in invertebrates and fish in the headwaters, so that transfer is occurring. But there is, as far as I know, in Northern California, the only data that exist show that when this experiment was done and added large numbers of carcasses, it had absolutely no effect on salmonid growth. There may be qualitative differences, but they weren't showing up in increased growth of the salmonids. Now, the belief in this is pretty strong. As you can see, here's an interesting procedure of grinding up huge salmon carcasses and spraying them out into the stream as a way to add nutrients. This is a little bit counterproductive to the idea that light is in fact limiting and not nutrients. particularly in alder-bordered streams. Red alder is a nitrogen fixer, and so a very good producer of nitrogen. So, but at any rate, you can see that this idea that we need to replace the migrating salmon nutrients that resulted when the adults die upstream needs to be replaced. My contention is that there are other management strategies, at least in the smaller streams, that would be probably more effective, and I think the work by Wilsbach and Harvey certainly demonstrated that. Okay, so what kind of questions should we be addressing? Because if we're going to use this management strategy, there are some things that I think we need to know before we can be fairly assured that these strategies are going to work. First of all, do the canopy openings change the stream and terrestrial invertebrate community structure? That is, if we make these manipulations, does it translate into those invertebrates most likely to appear in the salmonid diets? And assuming that would be what drives increased in salmonid production. Secondly, are these changes related to increased non-filamentous algal production or changes in the detrital pathways? That is, are these changes related to the R part or the P part of the PR ratio? Or probably both, maybe combinations, particularly with seasonal differences. Secondly, how are the changes in stream and terrestrial invertebrate communities related to increased salmonid growth? That is, which invertebrates are actually finding their way into the diet of the salmonids and producing the observed increase in salmonid growth? Well, one of the things that it seems to be coming through, as I mentioned, the collector group of invertebrates. These are the ones that feed on the fine particulate organic matter, which is generated by all sorts of processes I discussed earlier in the stream. The ones that seem to show up most regularly in the diets are the invertebrates that fall into a category we call behavioral drifters, as opposed to accidental drifters. The behavioral drifters exhibit a phenomenon of pulsed entry into the drift at dawn and dusk. The accidental drifters show no pattern of introduction. It's fairly random. It just depends on what physical characteristics of the stream may displace them at any time, not keyed to the diurnal cycle, but at any time. So this regular pattern of being in the drift at dawn and dusk, which of course corresponds with the major feeding times of salmonids, dawn and dusk, that enhancing that portion of the invertebrate community seems to have probably the most influence on changing the growth rate of the salmonids. Here's a figure that doesn't show up too well, but on this scale is the percent in the drift of behavioral drifters. On the y-axis here is a percent of behavioral drifters. On the x-axis is the growth rate, measured growth rate of salmonids in the stream, and you can see that as the percent of the behavioral drifters in the drift increases, so does the growth rate of the salmonids in those sections of streams exhibiting this. So this is only one set of data, but we have there are some other studies going on that seem to be showing the same thing. So that's the key. Most of these behavioral drifters are collectors, and it's quite predictable which ones we know now, which are the most abundant ones that show up as behavioral drifters and therefore in the diet of salmonids. One group of mayflies, the baited mayflies, probably are the most common and most abundant and most often encountered in trout stomachs or juvenile salmonid stomachs are in this category. So this certainly is a part of the invertebrate community we'd want to focus on with any kind of riparian management to make sure that group is in fact being enhanced. Okay. Just to come back to green diamond for a minute, Lowell Diller with green diamond and Matt House with green diamond and some other scientists, including myself, have been planning for some time, probably 3 years now, a set of experiments, and Lowell has certainly been the driver on this, And these experiments will be done on green diamond land. They will make extensive use of GIS information because a lot of the canopy issues can be measured remotely. Some of the temperature issues can be measured remotely. And then follow up on broader scale, larger scale manipulations of the canopy, both for amount of shading and species composition by selective harvest, using a sort of before and after approach with controls to actually try and measuring things in the stream about the fish, invertebrates, and in Lowell's case, amphibians as well. So it'll be a large-scale study over a long period of time, and as time goes by, the empirical database for the management will get better and better as we go along. Okay, thanks very much, and I'd be glad to take any questions.